Okay, guys, so who wants to go to Peru? Yeah, it looks fun, right? I think like if you haven't done much traveling before, then it's a good way to start, and then you'll get the bug, and then you'll have to always travel, and that's good. Yeah. Yeah, you know what? I tried to put that, but I had an issue. It'll go up after class. Yeah. Normally, I see you got spoiled. See, I used to always post it after class. No one ever complained. Now I post it before, and it's like, sorry.
One thing is that.
Okay. Yeah, sorry, I tried to, and it told me that like hackers were trying to get into my computer, and I was like, okay. And so I hoped it had actually gone up. But the hackers probably took it. I mean, I'm serious. And, like I was like, post, and then it was like, hackers are trying to get you. Buy our credit card. I don't know what they're get new insurance. It seemed all really weird and shady. But it'll be up right after this. Okay. So exam 1, if you took the exam on Tuesday, you should have your grade. If you took your exam on Thursday, it'll be up by Thursday. It's interesting. People have done really well. A lot of people got over 100, so that's good. There's some people who didn't do well, but there's always some people who didn't do well. So those of you who didn't do well, some of you have already contacted me. Please continue contacting me if you have any concerns. It does help sometimes to go over the exam with me, just so you can kind of see like what was going on. But one thing I have noticed is that people's mistakes tend to be because they were rushing and not reading the question fully, or they misunderstood it. So always call me over, because you can ask a lot of people in this class. They would call me over and say, I'm thinking you want this, is that right? I'm happy to say sure. Yes, that's exactly what I want. So that way you're not guessing on an answer. Like don't do it wrong because you don't understand the question. Get it wrong because you don't know it. Right? That's the reason. Yes? Once everyone's grade is posted, you can come see me and we can go through everything. You can look at everything. I just don't want the exam questions out there in the world. But I'm happy to meet you at any time to go over it. Email response time. I've been getting random emails from people, and I just want people to know, sometimes it might take me a little while to get back to you. I see your email. I got 2 or 3 emails about this last quiz. I saw them. I just have to have the time to get back to you. So usually within 24 to 48 hours, I will get back to you. Okay. Let's take 5 minutes to rate. So we've been starting since last week to talk about signaling, and we have so far just been very introductory, just talking about the concept of signaling and what generic signaling pathways look like, stuff like that, right? So take a minute here, just to kind of review what we've done so far. Signaling is usually initiated by a ligand binding to a receptor. Name 4 kinds of receptors and a defining characteristic of each that differentiates them. So usually when you're doing signaling, it's helpful to start to compare and contrast things so you can understand why I'm teaching you. So it's not just a laundry list. So this will be your first step. Start at the top, and we're going to work our way all the way down to the bottom. Okay. Hannah? Hannah? Okay, so you said ion channel receptors, G protein coupled receptors, enzyme linked. Yeah. What was the one more that's missing? Nuclear receptors. Yeah. Okay. Good. Good. Okay. So if you had to think of something to help to differentiate them, what would you say is a defining characteristic that is unique about the ion channel receptors? Oh yeah. No. Go. Where it's like, okay, so a signaling molecule is going to bind, but what's it going to do to that receptor? Activate it, but what's going to happen? Why is it called an ion channel receptor? Does it do something to an ion channel? Right, so it's basically going to open it, right? So ion channel receptors are the only ones that directly have a ligand bind to them and open an ion channel. Now, that's not to say it's the only ones that are associated with ion channels, because which receptor can regulate an ion channel? GPCR. But that's not a direct, it's not directly, the ligand's not directly binding and it's opening, it's through a different mechanism. So what can you guys say is unique about, thank you, Hannah. What can you guys say is unique about GPCRs that the other ones don't do? Yeah.
I said it works as like a molecular switch because it has the change between the GDP to the GTP.
Okay, so I hear molecular switch, GDP, GTP, right? So there's a G protein. So it's the only type of receptor that is, and it's a class. There's not just one GPCR. There's something like 700 of them, and they are involved with different pathways and different responses. But that family of proteins, GPCRs, are the only ones that are G protein associated and act as a molecular switch and are regulated by the GDP-GTP binding state. Okay, what was another one? We said enzyme linked, right?
I have a question about the G protein.
Yes.
Are they the only ones that indirectly regulate other membranes?
Are they the only ones that indirectly regulate other?
I know like an ion channel that would kind of indirectly.
Right. So exactly. So that is the only class. I hate to say only, but yes, that's the only class that we're learning about that indirectly is activating something else. Yeah. So you get signaling through your GPCR and something else. So not sometimes an ion channel is open, right? Compared to an ion channel receptor where your ligand is directly binding it. Yeah. What else? Enzyme linked, right? So what's unique to enzyme linked receptors? Do I have to call someone?
Instead of a single fast
So I just heard someone say single pass transmembrane. Well, that is a difference, right? GPCRs tend to be 7-pass transmembrane. And you guys should be able to tell me exactly where those proteins are translated and how they get into the membrane, right? Yep. Okay, I see some people nodding. Hopefully everyone knows. Yes?
It has intrinsic enzyme activity, like kinases.
So one kind is intrinsic. So it has either intrinsic or is associated with an enzyme, right? So it's a single pass transmembrane protein that has an enzyme activity within it, or is bound to a kinase, but it's usually kinase activity, right? Okay. What's the last one? Nuclear receptors. So what's unique about nuclear receptors? Yeah.
They're not on the cell surface.
They're not on the cell surface, right? And what else can you tell me about them that's unique that none of those other receptors do? Do any of the other receptors act as transcription activating proteins themselves? No, like as far as I know, none of the other ones will actually leave the membrane and go into the nucleus and bind to the DNA and activate gene expression, right? But nuclear receptors, that's what they do. They change their conformation when ligand binds and are able to go into the nucleus, bind to DNA or act as a co-activator, something like that to activate transcription. So that's unique. So it's useful when you're studying to kind of compare and contrast and think to yourself, how are these things different? And then try to attach those functions to the name so you can remember them. Yeah. They basically directly regulate transcription by themselves going into the nucleus and interacting with promoters and enhancers and like that. Okay, very good. Okay, so far we just spoke about generic signaling pathways. I gave you a few examples. We talked about different kinds of extracellular signaling molecules. And you might also want to start to think about, well, which kinds of molecules are going to associate with which kinds of receptors. We spoke about different kinds of receptors, and that's what we just talked about in our writing exercise, right? So these were so this is where we left off on Thursday. Right, so there were 3 kinds of cell surface receptors, and then that 4th was not cell surface, right? Ion channel, G protein coupled, enzyme coupled, and then there were the nuclear receptors. Right, so these are what we were just talking about, our ion channel coupled receptors. You actually have your signaling molecule interacting with the ion channel itself. And it's either closed or open based on the signaling molecule binding. So that signaling molecule binds and causes a conformational change, which will open or close the ion channel. And so this is often involved in signaling between nerve cells and stuff like that. GPCRs, and here you're dealing with a receptor that's interacting with a G protein. And it's going to activate some other protein or protein complex. One example was an ion channel. Another example is adenyl cyclase. And so then these were the enzyme-like receptors. So notice the difference here. 7-pass transmembrane protein interacting with G protein. Here, single pass transmembrane. And then there's either intrinsic kinase activity or it's going to bind to some kind of enzyme, often a kinase. But there are also other, like phospholipase C, there's some other enzymes that will interact with these receptors. But they either have their own enzyme activity or interact with an enzyme. Okay, so that's where we left off. And so I just want to remind you guys of this concept of molecular switches. Right? We talked about it at length in terms of GTP binding proteins, but just think of it more broadly in terms of NTP, nucleotide triphosphate. You can have GTP, you can have ATP. And with ATP, really what we're talking about is phosphorylation, right? You can get, and both of these are involved with signaling. Not only, so the GTP binding is not only RAB and RAN. This is a pretty broad mechanism for regulating proteins that are involved in binding to something and making something happen, right? We talked about RAB, we talked about RAN. Now we're going to talk about RAS before you know it. Right, so that's GTP. And then with ATP, you'll either have a protein will be off or it'll be on depending on whether or not it's phosphorylated. Now, this is kind of misleading because it's suggesting that the protein's only on when it's phosphorylated. That's not really the case. Just think of it as modification of activity. Sometimes things will be turned off when they're phosphorylated. Sometimes things will be turned on. Sometimes it's just creating a docking site for binding to something. But these proteins are really pretty important in signaling pathways. You're going to see tons and tons of kinases, and kinases are going to be phosphorylating other kinases, which might activate them, or might make a docking site for another kinase, or another protein, or some protein that's going to spread the signal to multiple pathways. And so this is just what you saw before with GTP and GDP. Right, remember we have our GTPases and the gaps regulate them. That's not unique to what we learned before. We're going to see it again. So the other thing to think about, just generally when you're thinking about signaling, is it's not always one signaling molecule binds to one receptor and activates one pathway. Often a combination of signaling molecules bind to a combination of receptors and activate one pathway or many pathways. It's a lot more complicated than just like signaling molecule, receptor, kinase, into nucleus, activation of something. It's good to start thinking of it in a very basic way, but then allow yourself to understand that it's a bit more complicated. So sometimes you'll have these integrating proteins, which right here is shown as this blue blob. And the purpose of that protein is to activate a unique signaling pathway when it's activated by two different receptors. So you might have receptor A and receptor B, which binds to signaling molecule A and signaling molecule B. And signaling through those receptors is going to activate kinases, which will phosphorylate this integrating protein. And only when it's phosphorylated by both pathways, you'll activate downstream signaling. And so just to make things a little more complicated for you. Does anyone have any questions about that? Yes?
Since the phosphorylation doesn't always mean that the signal pathway is on, but in this case, that is right.
Yeah, I mean, so yes. So don't always think phosphorylation equals on. Because I feel like that's a dangerous path to go down because sometimes it's turning something off. And so I always say, think about it as changing the shape. And so if the shape that's needed is to be phosphate bound and then SH2 domain interacting in order for signaling to happen, then it might be activating. But let's say in some cases you have phosphorylation, and we'll learn about this in the cell cycle when we start talking about that. Sometimes you'll have phosphorylation that's right in the active site of another kinase, and that's going to inhibit it, right? Okay. So here we're still very generic, right? So there's activation. You want to start from the top and think down when you start thinking about signaling, right? So we spoke about different kinds of receptors. And so when a receptor is activated, there's going to be some sort of conformational change or something that's going to induce phosphorylation or something is going to change that activates the pathway, right? And so then you'll get these signaling complexes that will form. And so you can kind of put them in three different categories. There might be recruitment of a signaling complex, a preformed signaling complex. What do I mean by signaling complex? What does that mean to you? Signaling complex. I'm saying a preformed signaling complex. Yeah.
Probably like a bunch of proteins that come together in order to communicate the signal.
Yeah, a bunch of proteins that come together to help to communicate the signal. Exactly. So, and they're regulated in different ways and they exist in different ways, but like what I was telling you the other day, when you're thinking about signaling, you don't get bogged down on individual pathways, just think about general themes because you tend to see them again and again. And so there's three main types of signaling complexes. There are these preformed signaling complexes on a scaffold that will become activated when ligand binds. There are, you can get assembly of a signaling complex at a receptor when ligand binds. So, and I'll show you some figures that that will make you understand this even better. And then you can also get assembly of signaling complexes on phospholipids in the membrane. They become hyperphosphorylated, and that creates binding sites for recruitment of signaling proteins. Right, so this is your preformed signaling complex. Right, so when there's no ligand bound to your receptor, you're still going to have some scaffold protein. Scaffold protein is just a large protein that has multiple binding sites for multiple signaling molecules. And so you'll have all this interacting, your scaffold protein and your different signaling molecules, and it's just there and it's just waiting. But when you get activation through ligand binding, then you're going to get activation of a kinase domain that will then phosphorylate things, which will maybe activate other kinase domains, and then you'll get activation of this complex, which usually really just means recruitment of other proteins that are going to then phosphorylate other proteins, with the end goal of changing the response of the cell, activating gene expression, changing the shape of the cell, inducing cell division, something like that. Yes?
Now, is this in a specific, one of the specific modes that we just talked about, or?
Yes, this is preformed, so this is, I'm just going to go in the order of that list. So this is the preformed signaling complex on a
No, I
scaffold.
mean, like, does all of these work in ion channels? These
Oh. So no. This is, a lot of these are going to be more associated with enzyme links. Because with the GPCR, so the question is this specific to different types of receptors? And I would say you're more likely to see some with different receptors. And a lot of these are going to be more associated with your enzyme linked receptors, because the way your GPCRs function is to activate something that's going to induce a flood of calcium into the cell from the ER, or a bunch of cyclic AMP into the cell by activation of adenyl cyclase. It's kind of a different mechanism. And then ion channel, well, that's just ion channel, right? It's going to just change the conformation, open the ion channel. So I would say you can think of these as mostly happening with enzyme linked or enzyme associated receptors. That's a good question. Okay. So the other one we just spoke about is sometimes you might have your receptor and nothing's going to be interacting with it, but once you do get your signaling molecule binding, it's going to phosphorylate itself and create binding sites for recruitment of signaling molecules, and that's what you see here. And so this is assembly of a signaling complex on an activated receptor. So that all those proteins are being recruited to the activated receptor because it's been phosphorylated, and that creates binding sites. So if you don't have activation, you don't have phosphorylation, there are no binding sites for these signaling molecules. That's how it's regulated. So another thing that you could see is where you're actually getting modification of the phospholipids in the plasma membrane, and that creates binding sites for signaling molecules. And so you'd have an inactive receptor, and you might have some phospholipid molecules just hanging out there, but they can't be bound by signaling proteins until they're hyperphosphorylated. And binding of signaling molecule to the receptor is going to phosphorylate your phosphoinositides and recruit, that creates binding sites for signaling molecules. Notice there's a theme, right? Activation, phosphorylation, recruitment of protein, right? It's kind of the same thing over and over again, but just slightly different. So then when you have recruitment of these proteins, they'll kind of signal along the membrane and then activate a signaling pathway. Any questions about these? Do they make sense? Are you wondering, is anyone wondering what this has to do with signaling? Okay, I just want to make sure. Good. Okay, so this is what I mentioned the other day with protein domains. Right, so there are these modular protein domains, and they have different functions. And they either mediate different interactions, bless you, or have different functions. Like an ATPase domain is likely to do what? Hydrolyze ATPase. I heard someone say that. And so there are some common domains that you find in signaling pathways. You'll find kinase domains, you might find ATPase domains, you might find GTPase domains. You'll find also a lot of SH2 and PTB domains. And those are different parts of the protein that are going to bind to phosphotyrosine. And so that's showing that right here. So here's your receptor, and it's been phosphorylated. And the part of this signaling molecule here that's interacting with the receptor, that's a PTB domain. And what it's specifically interacting with is a phosphotyrosine. Or there's this GRB2 adapter protein that's going to assemble on this protein here through its SH2 domain, and that's another phosphotyrosine binding domain. So remember, we're getting all of this phosphorylation, so that's creating all these phosphotyrosenes. And that's creating docking sites for proteins, and they're going to bind to specific domains. Two other domains that are mentioned in the textbook, if this is confusing, they write a nice little bit about it in the text, so I urge you to go read it. SH3 domains bind to proline-rich amino acid sequences. Proline-rich usually means there's a lot of terms in the structure. And so you can see here your SH3 domain is interacting with this proline-rich region of this other protein. And this SH3 domain is interacting with this proline-rich region of this other protein. And then there are also these pH domains, or pH domains, which bind to the that's bad spelling, phosphoinositides. So this is when you've got this hyperphosphorylation of your phospholipid in your plasma membrane. That's going to bind specifically to a certain sequence within these different proteins. And so I want you to start thinking about signaling as it's just pretty much proteins interacting with proteins, right? And they're going to interact with each other through basically whether or not the protein is phosphorylated or not. Or you might get activation of something based on whether or not something's phosphorylated. I just want to take a minute, though, to talk about the domains and that concept, because every year people get very confused, and on the exam I get an SH2 protein binds. SH2 does this, SH2 does that, and it's not a protein, it's a part of a protein. So when you've got this big, so each of these different colors is a different protein, and it's just the different bits of it have different names. And the reason they're called SH2 domains or SH3 domains is because they have similar structure that allows them to do a similar thing. Okay? And so I put in a link to this website. Let's see what we can do here. Maybe. And so it's just going to take you to this lab's website, hopefully. We'll see. Oh, see, how come this always does this? I do like the smallest thing, and it wants to make us nauseous. Let's hope this doesn't mess anything up. And so I wanted to really drive home the point of the function of a domain. Specifically, just the example I put here is the SH2 domain. And it's a sequence that makes a structure that's conserved. And that's why it's called an SH2 domain, because many different proteins have it. And so this is kind of showing the structure. And you're going to find it in many, many signaling molecules. And that allows for it to bind to phosphorylated tyrosine. Why would you want to bind to phosphorylated tyrosine? Uh-oh. Okay, go home. No, I'm just kidding. Okay, because that's the whole thing. If you don't have the the appropriate proteins recruited to the right proteins for the signaling cascade to continue, you're not going to get a response to anything, right? And so by having phosphorylation happen, then you get recruitment of a protein to that phosphotyrosine that's going to relay the signal to the nucleus and activate gene expression. Okay? And so this is kind of showing, so I want you to go to this website and just fool around a little bit because this lab has a really nice kind of library of different domains and how they look. And it's showing here this SH2 domain is found in many different types of proteins. And you can make the assumption automatically that these different types of proteins bind to phosphotyrosines. So it gives you a list. There are adapter proteins that are going to have SH3 and SH2 domains. What was SH3? That was binding to proline-rich regions. Was that right? There's scaffold proteins that have SAM domains. I didn't tell you about those, but those exist. There are kinases that are going to have a kinase domain. Duh. Right? Kinases should have kinase domains. Does everyone agree with me? Okay. RAS signaling that has a RAS gap domain. That means it has the GTPase domain, right, for hydrolyzing GTP. Transcription factors. Some of them have SH2 domains, and they're also going to have a DNA binding domain. So I just want you to think about, I don't know, I found that once I understood proteins in the sense of like they have these different parts that do different things, everything started to make so much more sense. So I'm trying to push that there. Okay, so take your time to kind of look through this website and read and think, and come see me if you have questions about the concept of domains, because I'm telling you, every year. I went to Dutch Wonderland this weekend, and there's this ride that you go on that this is like the house spinning around. Oh my God. The person who was running the ride looked at me and was like, "Do you feel sick?" Like, looked at me immediately, close your eyes. So I'm like, the kids were like, "Woo!" And I'm like, that's how I feel, feeling like I was lecturing. Spinning. Okay. So here's a quick practice problem to see if you can apply this concept. Okay, so you're studying a newly identified protein from a virus that infects birds. You have the protein's amino acid sequence, so since you don't know what it does, you decide, well, let's blast it and see what it's similar to. So you run a blast search and you get a whole bunch of hits, and one of them that's returned has 98% identity. It's a protein that has 98% identity to your unknown protein, and it has a kinase domain, so it has kinase activity, and it has an SH2 domain. So what hypothesis can you form about your unknown protein? So what do you think, based on the fact that it has this similarity to a protein with a kinase domain and an SH2 domain, what do you think your protein might do? What cellular process might it participate in? And how would you experimentally evaluate this? So go ahead and discuss with your friends for a minute, and I'll circulate and make sure you understand the question.
And it can either yeah. Then all the so and then my country stuff. Right. Yeah.
people are still talking about this, and how many people are talking about what they did last weekend? You already know I went to Dutch Wonderland. From the dream. No. Actually, Dutch Wonderland, I have nothing but positive things to say about it because 3 out of 5 of the tickets we had to buy were free. And I'm happy I had to buy 5 tickets. I would have had all bad things to say about it. It's expensive. A lot of money to go on rides. But at least, one thing I did notice is when you go to the fair, there's lots of rides, and if you don't know that you want to go on them, you see the guy who's pressing buttons. Because you're like, okay, this guy lives a rough life. I don't know how he's doing today. Who expected this, right? But at Dutch Wonderland, it's like, everyone's so wholesome. They all look like they slept in bed the night before, ate a good meal. So you feel a little better putting the love of your life on this ride and walking. Anyways, let's talk about this problem. Okay, so, problem? Am I right? Yeah. So you had a question. Do you want to share the question with the group? Right, okay, so you're like, okay, so it has identity with a protein that can phosphorylate and a protein that binds to phosphotyrosine. So what do I do with that? Right, that's your question. How many people felt that way? Okay, yeah. I mean, it's hard to know where to go until you've done it, right? So yeah, so you started with a protein that you knew nothing about, right? And so then you really have nowhere to go, right? It could do anything, you have no idea. But by comparing it to other proteins and seeing that it has similar domains, you can start to form a hypothesis because you're like, well, it's similar to this protein that has kinase activity. Maybe it's a kinase. It's similar to this protein that has an SH2 domain. Maybe it binds to phosphotyrosines. It's similar to and usually when you blast something, you might get 50 hits. And you might find that it's similar to 10 different proteins, all that which have SH2 domains and kinase domains, and then you look at those proteins and say, oh, these are all involved in signaling. And so then you can hypothesize, maybe my protein, my unknown protein, can phosphorylate things and bind to phosphotyrosines and participate in signaling. And that's how you would form your hypothesis. But then the question is, how do you evaluate whether that's the case? Because just because it's similar to something doesn't mean it truly is. Yes? So you're saying compare a wild type version of your protein and a mutated protein where you've removed the SH2 domain or the kinase domain and see how it functions, right? So if you remove the SH2 domain, what would you be looking for? Effect on what function? Right, it shouldn't be able to bind to phosphotyrosine anymore, right? And if you remove your kinase domain, what are you looking for? I just heard phosphorylation. Okay, yeah, so it shouldn't be able to phosphorylate. You always need to compare it to your wild type. Your wild type should do these things, and then your mutant shouldn't be able to do these things. And so if you do those experiments and you're like, oh, okay, so it truly does have a kinase domain, and it truly does have an SH2 domain, then you can think, well, maybe it does function in signaling. And so then I got a comment while you guys were working on this that was like, well, can you tag it and follow it and see where it goes? Maybe you can start to see what proteins it interacts with. And then based on what proteins it interacts with, you can start to figure out which signaling pathway. So let's say you fluorescently tag your protein, and you look at where it is in the cell. And let's say it's at the plasma membrane. Then you might say, oh, maybe it associates with a receptor, right? Because those are the plasma membrane. You can see what it interacts with. What if it interacts with RAS? say, oh, well, let's look at signaling pathways that are involved with rest. And why would the viral protein want to interact with the signaling pathway? Why? So it can get into DNA into the nucleus, so it can force the cell to do what it wants it to do, right? Sometimes you might need to activate a signaling pathway to push a cell into cell cycle. And that will help have all the things available for replication of a viral genome. And so a viral protein might interact with a signaling pathway to kind of force the cell to do that. Any other questions about this exercise? Do people understand now that we've explained it? Okay, if not, continue. Yeah.
So what's the formal hypothesis?
The hypothesis. So who wants to answer that? What is the hypothesis that we formed? Yeah. That the protein is probably involved in signaling. Or more specifically, that it what? That's involved in signaling complex, but I want even more specific, because a hypothesis needs to be testable. And so the hypothesis that it's involved in signaling is not very testable, right, with one experiment. It might be the culmination of 10 experiments. Yes? That the protein is a receptor. That you can form that hypothesis. But what about the things that we spoke about with the kinase domain and SH2? Yeah.
The proteins involved in gene expression.
The proteins involved in gene expression. These are all hypotheses that are very large. Get more specific. Yeah.
That the protein is involved in kinase activity and possibly binds to phosphotyrosine through the SH2 domain.
Great. So that the protein has kinase activity. That's very testable, right? Either you can phosphorylate things or you can't. SH2 domain, either you can bind to a phosphotyrosine or you can't. Very, and then these other broader hypotheses are very important hypotheses to have because they're going to help direct you to where you need to go. Because no one's going to publish a paper that's like, protein X has a kinase domain. No one cares about that. But really, when you're doing research, it's all about publications. And you have to have a story, and your story might be participates in signaling, is a receptor, all these things, and then in the end you've got this amazing story where you're like, this bird virus interacts with the, I don't know, MAPK, blah, 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 signaling pathway, and forces the cell into cell cycle. And then, woo, that's cool, that's the story. It's like, yeah. Signaling is pretty awesome. I promise you, you'll think that. Okay. So we were talking about signaling. We're talking about turning on signaling through a ligand, binding to a receptor, and then you get assembly of a signaling complex, and there's phosphorylation, and there's interaction with proteins. All this happens, right? But don't forget, when you turn on signaling, you also need to turn it off, right? Because if you keep signaling on, well, what's the point of having an inducible system if you can't turn it off? So you can induce it again when it's needed, right? You don't want to keep signaling on. And so these are 5 different ways that you get desensitization, or basically just turning off signaling through targeting the receptor, right? These first two should be very familiar, right? Because we just learned all about it for the last exam. We talked about endocyte synthesis, right? We talked about recycling of receptors. We talked about sequestering receptors. This is actually a very incorrect figure, isn't it? Shouldn't that be something else? A multivesicular body, exactly. Right? Because what's to stop the cytoplasmic tail from signaling within the endosome? That can happen, and that's why they do butt into multivesicular bodies. I looked at the most recent edition of the textbook, and they still haven't updated that figure. It takes, I think, that's fairly recent. I don't know, because they did talk about it in the textbook. I don't know. So someone's got to call the editor. Tell them. You call the editor. Tell them to update the textbook. The other one is endocytosis and going to the lysosome, right? Degrade your receptor. Does it make sense how degrading your receptor might turn off signaling? Pretty straightforward there. The other thing you can have is receptor inactivation. So maybe a protein will come and bind the cytoplasmic tail of your receptor and prevent interaction and assembly of that signaling complex, right? And that can be different ways, right? You can get, for GPCR, if you kind of hide the part of your GPCR that interacts with your G protein, you're not going to get any signaling because then it can't activate your G protein. Or if you hide the part that's going to get phosphorylated and bind to signaling molecules, that's also going to turn off signaling. You could also get inactivation of intracellular signaling proteins. So sometimes receptors will activate other proteins and you can inactivate that. That will kind of cut off the signaling cascade lower down. And then a lot of signaling pathways produce inhibitory proteins. What's that called when a pathway produces an inhibitor? Something feedback? Negative feedback. Negative feedback. So that's when you turn something on and it makes its own inhibitors shut itself off. The NF-kappa B pathway does that all over the place. And that's important, right? Sometimes when you activate something, you want to produce your inhibitor so that the signaling is very short-lived. That you have enough inhibitor there that can shut it off quickly. Questions about this? Okay, so let's talk about GPCRs. Here, so these are those 7-pass transmembrane proteins that interact with the G proteins, right? Automatically, you should start to think certain things. GPCR, 7-pass, interacts with G protein, GTP dependent, activates other things, right? Yeah, sure. People are getting sick. I'm so mad about it. You guys get mad when you're getting sick? You're just like, body, why are you fooling me? I don't have time to get sick. Anyhow, at least it's not Ebola. Oh, yeah. You need to get positive. Right? That's a bad one. Oh, God. I just feel for the whole situation. It makes me so sad for people. I went to take my son to the pediatrician yesterday, and they're like, have you been out of the country? I'm like, we don't have Ebola. She just laughed at me. But like, they're all everyone's screaming, everyone's freaking out. I blame it on the media, right? It's always the media's fault. I mean, it is a nasty virus to die from, but it's very difficult to get. Anyway, GPCRs, right, 7-pass. Sorry, I'm a little distracted today. Okay. So activation of a GPCR results in a conformational change and activation of the GTP binding protein. So what does that really mean? Conformational change kind of allows GDP to be released. And then there's GTP present just in the area, and so then it can bind. And so you can think of an activated GPCR as kind of like a GAF, right? What does a GAF do? Changes from GDP to GTP, but more specifically, what it does is it allows for release of GDP and allows for binding of GTP. It's all related, isn't it? That's why if you don't get this concept in the beginning, hopefully you get it by now. Right, so this is the structure. There are three different subunits: alpha, beta, gamma. Very novel naming. And you're going to have a GDP binding site, and when you can kind of imagine when you have a conformational change, you're not going to have strong binding with GDP anymore, right? There's going to be an ideal conformation for GDP binding, and so when you get signaling through the receptor and a conformational change, it's going to change, and so now you won't have that tight binding with GDP, and that's what releases it. Questions? Comments? Actually, I don't want to hear any. Okay, right, so you'll have your GPCR and then you've got your inactive G protein. And when a signaling molecule binds, there'll be a conformational change. And then GDP is released and GDP is done. So now it's active. So what? Right? Yeah, so it's active. Yay. What's that have to do with anything, right? Well, really, it's going to activate a whole signaling pathway. So one of the things it'll do is it'll induce production of cyclic AMP. And so this is out of the textbook, and it's showing cyclic AMP levels in the cell. And so at time 0, cyclic AMP levels are fairly low. And when they add serotonin, which is the ligand for the GPCR they're studying, more red is higher levels. They're using some fluorescent protein that is more fluorescent and it binds to cyclic AMP. So within 20 seconds, tons of cyclic AMP. A fairly quick response. And so I usually think of GPCR signaling as very rapid. I don't know if that's a good thing to be thinking, but that's just how I think about it. So there are a lot of different responses that are often hormone-induced that are mediated by cyclic AMP. And I remember learning this and being like, okay, cyclic AMP, I don't get it. And so what made it all make sense to me is understanding that cyclic AMP is going to bind to something and activate something, right? And so all that cyclic AMP is induced, and what it does is it activates PKA, which is a kinase. So it's basically turning on a kinase. So normally you'll have this inactive catalytic subunit, and once cyclic AMP binds to the regulatory subunit, it releases from the catalytic subunit, and you get activation of the kinase. And so what does this have to do with anything? So if you look at the whole pathway, this is just one example of a cyclic AMP regulated pathway. You'll have your GPCR, which is bound by a signaling molecule, so then you'll get your conformational change of your G protein, right? And it'll activate something. And in this case, it's going to activate adenyl cyclase, which is what makes cyclic AMP. And so you get this huge amount of cyclic AMP, which will then activate your kinase, PKA. And when it's active, it can go into the nucleus, and it can phosphorylate a regulatory protein and activate gene expression. So that's the big picture. Big pictures it can do when you're looking at a signaling pathway. But this whole thing, so like let's say you call this serotonin, and then you get all the cyclic AMP, and then you get activation of gene expression. What it's really doing is activating expression of genes that are associated with responsive neurotransmitter. Right, so this is just a close-up. Signaling molecule binds to GPCR, activates the G protein, activates the adenyl cyclase, which makes cyclic AMP, which activates PKA, and now PKA can go into the nucleus and activate CREB, which then can bind. You get phosphorylation of CREB, which can bind. It's a gene regulatory protein that activates expression of these genes. And there's a specific binding site on the DNA for CREB in this case. And so when this happens, CREB can go in and bind to all the different genes that have this binding site just upstream. And then what that does is that recruits RNA polymerase and all the general transcription factors and RNA polymerase, and then you get activation of gene expression for that subset of genes that has this binding site. Any questions about this? Okay, I think we're going to stop here for today, and we'll continue on